Hello, welcome to lesson 7.4. Today we'll be talking about bacterial feeding nematodes again, particularly in their relationship to bacteria. So we'll look at the nematode-bacteria interactions or relationships and how they select for particular species or groups of bacteria, and also about how the soil habitat in general in terms of nutrient content, as well as soil structure and the other organisms that are present in the soil affect bacterial feeding nematode populations in various soil ecosystems. Okay, so let's jump into lesson 7.4. Just like us, each organism, including nematodes, actually selectively choose their foods. They don't all love the same foods, and they all are specifically structured for specific foods. But some species of nematodes harbor specific symbiotic bacteria in their guts, on their cuticles, and this affects bacterial taxa abundance and changes the composition of the soil bacterial community, because nematodes can actually transport and deposit different bacterial taxa, sometimes in places where these taxa are not present. Nematodes can protect and disperse bacteria into favorable environments for those bacteria, so that actually almost means that they are farming these bacteria by dispersing them and taking them to new habitats and grazing on them and increasing their populations. Selectivity also depends on whether the nematode can sense bacterial odors that the bacteria produce and therefore entice the nematode as a food source, just like we might like the smells of different fruit. And those fruit smells entice us more than other fruit smells. Also, the size of the bacteria and the shape of the bacteria matters. If the bacteria is too big for a nematode to fit it through its mouth, then of course the nematode won't feed on that bacteria. But also the shape of the bacteria is important. Filamentous bacteria are not readily eaten by bacterial feeding nematodes, as they need to suck in the bacteria through their mouth parts. However, sometimes there's specialized feeders that can exploit a niche of bacteria that others might not, depending on how their mouthparts are shaped and the signaling between the bacteria and the nematodes. Some bacteria produce biofilms and pigments, as well as polysaccharides and toxins that actually deter nematodes, or nematodes cannot digest them. They might be able to consume them, but then they have to excrete them. And in terms of toxin production, well, obviously the nematodes won't want to eat a toxic bacterium. And these toxins might work on some nematodes but not others. So you get the picture how varied this is and how dependent it is on the specific food characteristics for nematodes. The feeding apparatus of the nematode and the chemical cue detection provide constraints in terms of size of the mouth, as well as the ability to detect bacterial odors, as said earlier. Interestingly, even closely related species of nematodes may prefer different bacterial strains to consume. So it is a very specific and therefore selective grazing scenario. Selective grazing alters bacterial community. Example, an increased abundance of proteobacteria through the process of enhancing these bacteria to grow alters carbon mineralization. So this is just a specific example of how these interactions actually have huge effect on the soil ecosystem. Selective grazing also decreases microbial metabolic quotient, and this is the microbial respiration rate per unit of microbial biomass, which determines soil organic matter utilization capacity of bacteria. So it determines the ability of bacteria to utilize soil organic matter. So this reduced microbial metabolic quotient leads to soil organic carbon accumulation within macroaggregates. So once again, the effect of grazing and that specificity or selectivity affects soil organic carbon accumulation in soil. It also reduces the metabolism of soil organic matter, and that increases soil organic carbon accumulation in macroaggregates. So really, this is all showing us just how specific and intricate and also important these different interactions are between bacterial grazers and their prey, and how selective grazing affects the soil ecosystem and in fact affects soil carbon accumulation and therefore climate. It is quite intricate and quite magnificent in terms of the design of the system and its multifaceted effects on the entire soil ecosystem. And note the beautiful image by Reggie T of a nematode here. And so these nematode-bacterial relationships do become quite involved. So in general, while predation increases bacterial community composition and bacterial biodiversity, there's also this effect of new ecological opportunities being formed by the grazing mechanism. For example, the predator resistance strategies of bacteria is influenced as different nematodes feed on different bacteria. And also the nematodes, by default, by existing in the soil, by defecating and moving through the soil and disturbing the soil, they create predator-free spaces that increase bacterial diversity. And this is particularly true for when microaggregates are formed as a result of nematode activities. Interestingly, the bacterial communities in the soil, in the bulk soil, differ to those found in the nematodes themselves. And those bacteria that are in the gut of the nematode or on the skin of the nematode or living within the nematode can also be released in places where those bacteria may not have existed before. And so therefore, as the nematodes move throughout the soil, they actually disperse these specific unique bacterial species that they harbor. One study showed that the genus Pseudomonas and the family Symbionts were the most common bacterial taxa in all nematode feeding groups. They looked at the different species or taxa of bacteria in the different types of nematodes. But the relationships between bacteria and nematodes can be beneficial, and that means mutualistic, where both partners benefit, or actually harmful, where the bacteria are actually pathogenic or parasitic within the nematode. They can also be facultative, so that means that the bacteria can only survive within that particular nematode, and it cannot live outside of that nematode. That's where its habitat is, and that is its only habitat. And they can be stable and long-term symbioses, where over time that species of bacterium is associated with that species of nematode, or temporary interactions where there is only a short period of time where the bacteria are associated with the nematode at specific times, but not at others. Some of these relationships are extremely specialized, and the nematodes only select for specific bacterial genera or species, and these bacteria are then purposefully introduced to other environments as the nematode moves into new environments, and sometimes the nematodes will actually cultivate these bacteria or rear them. And it is very much exactly like our gut, filled with our own bacteria, depending on the particular foods we eat and the particular lifestyle we have. It's all very specific. This beautiful image is that of a green fluorescent protein labeled nematode from Japan. And I love just the different microscopy techniques that are available these days to demonstrate the beauty and the science in studying different organisms, and in this case, nematodes. To demonstrate just how specific these symbiotic relationships between bacteria and nematodes can be, a good example is that of entomopathogenic nematodes. These are nematodes that actually feed on insects, and in this case they use bacteria to actually kill the insect for them. There are at least two specific genera of nematodes that engage entomopathogenic behavior and particularly involve gamma proteobacteria in this symbiosis, where the infective stage of the nematode harbors intestinal bacteria, and these bacteria are key in the acquisition of the food source for the nematode. When the nematode that is colonized with these bacteria invades the insect host, the bacteria cause death of the insect. They help to degrade the insect to provide food for the nematode and also provide protection against any other opportunistic bacteria that might come along and want to consume the insect. So once the insect is killed, and in this case it is a larva of a beetle that's getting consumed, there are 4 to 5 life cycles of the nematode within that one dead body of the insect larva. And this is all happening in soil, of course. And a lot of these insect larvae are actually considered to be pests of crop plants. And so this is why this is so widely studied, where these nematodes can be used to actually control these insect larvae from consuming roots and therefore causing crop devastations. So within those 4 to 5 life cycles, the food resource usually is depleted and the colonized infective stage of the nematode finds a new insect host and the cycle starts again. And if you wanted to have a look at more closely as to how the process occurs, there is a reference to the study that shows this cycle. Just to clarify some of the acronyms, so EPN is entomopathogenic nematodes, and also the infective juveniles in this diagram are labeled as IJ. This diagram describes the initial stages of the maternal transmission of the symbiotic bacteria, and it begins with a stage when the infective juvenile exits diapause. So diapause is when animals, like for example nematodes, are able to enter a state of suspended development to survive adverse conditions. Like for example, when food runs out, they can survive this period of time, and then when food is available once again, the nematodes can resume their metabolic function. When these juveniles that are infected with the bacteria come out of diapause, they regurgitate the symbiotic bacteria and become completely free from this symbiont before reassociating with the symbiotic bacteria later on during their development. So the reassociation of these bacteria results in the formation of a persistent bacterial biofilm in the posterior intestinal cells in the nematode. And you can see the green color in the nematode showing the bacteria. So basically, in this case, the female nematode specific cells in the intestine are colonized, and she passes on this bacterial inoculant onto her offspring. So the juveniles will end up harboring these symbiotic bacteria in their gut as juveniles infect insects to consume. So yes, this is a good example of that very intimate, obligate symbiosis between nematodes and the bacteria that they harbor and use in order to gain a food source, and in this case, insect larvae. The soil habitat within which nematodes are found is very important as to their ability to survive in that habitat, their ability to proliferate and to breed. And that habitat depends on many factors such as the nutrient source for the food source, and in this case bacteria that the nematodes feed on, but also the structure of the soil. How does that affect nematode numbers, nematode diversity, who can actually live in that soil, as well as the predators that might be present there, like for example earthworms that will consume by default, being so large, nematodes as they travel through soil. So one study has shown that in soil that is subjected to conventional fertilizers, nitrogen, phosphorus and potassium, so NPK, when straw and pig manure were added, this affected bacterivore, omnivore and predator abundance and diversity. So basically having extra organic matter here as well as biochar that was added actually produced a greater abundance and diversity of nematodes in that soil. Another study showed that when rice residues were added to the soil, this increased bacterial feeders. However, when soybean residues were added, which contained twigs, this enhanced fungal feeders because there was more food now available for fungi to feed on, so that affected the fungal to bacterial biomass ratio in the soil, and therefore the nematodes that were able to predate on these different microbes. This study also found that when the organic residues were buried, as opposed to being left on soil surface, this increased nematode populations, which makes sense because the organic matter was now feeding the microbes within the soil, and therefore nematodes could take advantage of those increased microbial populations. Interestingly, the structure of soil is super important as well because microaggregates contain greater bacterial abundance and diversity, but less nematodes compared with macroaggregates. So basically, microaggregates contain less nematodes because the nematodes can not fit a lot of the times into the microaggregate pores and actually consume the bacteria present in there, in those macroaggregates. So macroaggregates, where the pore size is greater, the bigger nematodes can actually fit in through those pores and take advantage of that habitat that has specific bacteria living within those macroaggregates. So predation activity actually increases in macroaggregates, and that makes sense because once again, these larger nematodes fit in and feed on the bacteria, whereas the pore size in microaggregates reduces predation pressure on bacteria because it excludes the larger species of bacterial feeders that simply cannot fit into the microaggregates to forage on bacteria in those. There are also earthworm effects. So some studies show that earthworms will reduce the number of nematodes in the soil, and some that will increase the biodiversity and number of nematodes in soil. And that's got to do with the fact that earthworms actually feed on nematodes because they feed on all the organisms that surround the organic matter in the soil, and they have large mouths, and nematodes will end up undoubtedly in their gut. And at the same time, as they defecate and make their casts or castings, that is full of nutrients as well, which will increase bacterial activity and therefore attract nematodes. So there might be more nematodes, a stimulation of nematode populations as a result of worm castings. But if the earthworm populations increase to a certain point where that limits nematode numbers, then of course that has a negative effect on nematode populations within that soil. It just really depends, and various studies are showing various effects of earthworms, and that of course just depends on the habitat and many factors within that habitat as to what the dynamics between earthworms and bacterial feeding nematodes will be. Omnivorous and predatory nematodes also regulate bacterial feeders, so there's a feeding pressure on the bacterial feeding nematodes in the soil habitat, and that all depends on so many different factors that we've just mentioned above as well. So as the last image for this lesson, this is a beautiful photo by Eric Lundquist, and it shows a fluorescence label in a bacterial feeding nematode. It's a beautiful image, and it also shows some of the anatomy of the nematode. So we've come to the end of lesson 7.4 on bacterial feeding nematodes, and our next lesson will focus on fungal feeding nematodes. So we get to learn about the interaction of nematodes that prefer to consume fungi and are structured in a way that they can consume fungi. Okay, so you'll be hearing from me again in lesson 7.5. Bye for now.